Welcome back, students. Today we are going to discuss part 1 of Unit 9, that is application of biology. Before we start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In this particular video lecture, we are mainly focusing on the microbes used in household products and microbes used in industrial product. Okay, so these are our main points which we are going to discuss in this particular video lecture. So as you know, bacteria, virus, fungus, protozoa, they all comes under microbe community. So microbes are diverse in nature. There are so many microorganisms. Some virus, viroids, also prions are proteinaceous in nature and act as an infectious agent. And in this particular picture, you can see various shape of the microbe. This is rod-shaped bacterial structure. This is spherical, and this is the zoomed one. At 50,000 magnification, you can see this is a rod-shaped structure with so many fungal, so many flagella. Okay, so and now this slide is showing some viruses like bacteriophage is a virus, adenovirus, which act as a main respiratory infectious virus, some TMV virus, which is a plant virus. Okay, so this one is a plant virus, tobacco mosaic virus. This actually infect the respiratory tract of mammals or animals, and this is a bacteria. Bacteriophage yani bacteria killing virus. So different type of viruses, different type of bacteria, different type of fungal strains, they are mostly infectious in nature. Right, now let's understand bacterial and fungal colonies. As you can see, there are two petri plates. This petri plate is having so many tiny circular structure. These are known as colonies of bacteria, okay, or bacterial colony. This media contains, along with agar, some nutrient content, yani ki nitrogen, carbon, and other important source which is required for their growth. Right, and similarly, this is a fungal colony. You can see what is the difference between these two. Okay, this is a fungal colony growing on a petri plate. So microbe like bacteria and many fungi can grow on nutrient media and form colonies. And these colonies we can see with naked eye. Okay, and such culture are useful in study on microorganisms. If we want to study any effect of antibiotic or heavy metal or any substrate or whatever our target, we can just put that particular thing into the media and spread that particular bacteria. And this is how, with the help of a control plate and a test plate, we can compare what is the effect of that particular antibiotic or heavy metal on that particular bacteria. So this is how we can study bacterial strains or fungal strains in laboratories. But in household products, some microbes mostly are infectious in nature, but some microbes are useful for us. They have useful in our household products. They are useful in industrial way also, which we are also discussing later in this particular lecture. So first, some bacteria and their useful quality for as a household product. First is lactic acid bacteria. Lactic acid bacteria is actually convert milk into the curd. During growth, lactic acid bacteria produce acid, which actually coagulate and partially digest the milk protein. And this is how Lactic acid bacteria help us to convert milk into the curd. So for this particular purpose, a small amount of curd is added to the fresh milk as an inoculum or as a starter, and that small amount curd contain millions of lactic acid bacteria. Okay, millions of lactic acid bacteria, which at a suitable temperature multiply and convert milk into the curd. Okay, and it also help to improve its nutritional quality by increasing vitamin B12. So this is beneficial for us. In our stomach too, this lactic acid bacteria play a very beneficial role in checking disease causing microorganisms. Okay, lactic acid bacteria. Second use as a cheese. Cheese is one of the oldest food item in which microbes were used. Okay, different variety of cheese are known for their characteristic texture, characteristic flavor and taste. Okay, like in these two pictures, this picture, this this picture is having so many big holes. That's why we can use the term Swiss cheese for the same. And these large holes are due to the production of large amount of carbon dioxide by a bacteria named Propionibacterium sermini. With the help of this bacteria, we can produce this type of Swiss cheese. Another type of cheese is having green color inside it. And this type of cheese is known as Roquefort cheese. Okay, and this type of cheese are ripened by the growing a specific fungi on them, which give them a particular flavor as well. Okay, so for a particular flavor, sometimes some companies use that particular specific fungi to ripen up this Roquefort cheese. Okay, third used in household product is the dough or idli sambar batter. This is used for making food such as dosa, idli, and also fermented by some yeast and bacteria. And this puffed appearance of our dosa or idli is due to the production of carbon dioxide. Okay. Next, a number of traditional drinks and some food are also made by fermentation of the microbes. Okay, like a very popular drink known as toddy. It is a traditional drink of some part of the southern India, is made by fermented sap from palm. Okay, so toddy is a fermented sap from palm, and for the fermentation purpose, some kind of bacteria is helping out. Similarly, the dough which is used for making bread. Okay, so here baker's yeast or Saccharomyces cerevisiae is mainly used for the fermentation purpose. In the puffiness of the bread, or this large hole is again produced because of the production of large amount of carbon dioxide. Okay, so these are some examples in which bacteria help us in household product. Now let's discuss microbes in industrial products. Okay, so in industry, microbes are used to synthesize number of products that is very, very important, that play a very important role in human life. Okay, these are very, very valuable for us, like various kind of beverages, various kind of antibiotics are some examples. Production on an industrial scale require growing microbes in a very large vessel. As you can see in this picture, these large vessels are known as fermenters. So on industrial scale, fermenters are used for the production of different kind of beverages, various kind of antibiotics. So microbes, especially yeast, has been used from time immemorial for the production of beverages like wine, like beer, whiskey, brandy, and rum. For the production of any kind of alcohol, mainly yeast strain, different type of yeast strain is useful. Okay, so Saccharomyces cerevisiae is also used for the fermentation of cereals and fruit juice to produce ethanol. Which strain? Saccharomyces cerevisiae. We have to learn this. Wine, beer are produced without distillation. Okay, whereas whiskey, brandy, and rum are produced by distillation of the fermented broth. Okay. Now, antibiotics. So, antibiotics again, these are produced by the microbes. Okay, and the discovery of antibiotic is a main significant discovery of the 20th century and have greatly contributed towards the welfare of human society. Antibiotics. Anti means against and bio means life. So against life, not us. Again, this term is in context of the disease causing organism. Okay, with reference to human being, they are pro-life, not against. Okay, so antibiotics are chemical substance which are produced by some microbes and can kill or retard the growth of other. What are antibiotics? Antibiotics are chemical substrates which are produced by some microbes and can kill or retard the growth of other. Clear? Now, okay, now let's discuss some points related to antibiotics, as there is a very famous scientist named Alexander Fleming. Okay, he actually first time observed the Staphylococcus could not grow around a mold, that is Penicillium notatum, growing in his unwashed culture plates. From there, he extract this penicillium. So, first time isolation of penicillin, this particular thing, isolation of penicillin or discovery of penicillin is done by Alexander Fleming. In his laboratory, he mainly work on various microorganisms, and suddenly he observed that because of the presence of some mold, his microorganism, mainly Staphylococcus, is not able to grow in that particular plate. So he start analyzing what kind of microorganism is there, what kind of fungus is there, which actually inhibit the growth of a bacteria. So he extracted antibiotic, this penicillin from that particular plate. Later on, Ernest Chain and Howard Florey established the full potential of this antibiotic, penicillin. So for his significant work, Alexander Fleming, Chain, and Florey was awarded the Nobel Prize in 1945 for this very, very important discovery. Okay, because of the penicillin in World War II, doctors are able to treat American soldiers. Okay, so antibiotic are the chemical substance which is produced by some microorganism to kill or retard the growth of some pathogen. They are also useful to treat some deadly disease like plague, like whooping cough, like diphtheria, like leprosy. Okay, so discovery term is related to Fleming, but the term antibiotics, this term is given by Salman Waxman. Please don't confuse this. The term antibiotic is given by Salman Waxman, but discovery is related to Alexander Fleming. Penicillin antibiotic or first antibiotic is discovered by or observed by him along with two other scientists, but the term is given by Salman Waxman. Now, chemicals, enzyme, and other bioactive molecule which is very frequently isolated from various bacterial and fungal strains. So these are some bacteria and fungal strain name like Aspergillus niger. Citric acid is isolated from this Aspergillus niger. Acetobacter aceti is this particular bacteria is responsible for the production of acetic acid. Clostridium butylicum, this is again a bacteria from which butyric acid has been isolated. Lactobacillus, again a type of bacteria from which lactic acid has been isolated. So various strains of bacteria and viruses, or not virus, fungus, bacteria and fungus are used for the isolation of various organic acid, various alcohol and enzymes as well. Okay, like for alcohol, yeast is mainly responsible and Saccharomyces cerevisiae. This is used for the commercial production of ethanol. Similarly, for the enzymes like lipase, this lipase enzyme is useful in detergent formulation, which helpful for the removing of oily stains from the laundry. This is the picture. This contain lipase. Okay, some pectinase and protease enzyme are also useful to clarify bottled fruit juice. Bottled fruit juice. Normally, if we try to extract juice from the fruits at our home, that fruit contains sulfurous content also. Okay, so that contain pectinase and protease. So, bilkul clear juice which we got from the packed food, which we have purchased from different brands, that they actually use pectinase and protease to clarify their fruit juice. Okay. Similarly, another type of biomolecule or enzyme known as streptokinase. This enzyme is again produced by Streptococcus, and this enzyme used as a clot buster for the removing clot from the blood vessels of the patient which have undergone myocardial infarction leading to heart attack. Okay, so this is again a very important enzyme which is used as a clot buster. Clear? Now another antibiotic, cyclosporin. Cyclosporin A. This is produced by a fungus, Trichoderma polyosporum. Trichoderma polyosporum. And this cyclosporin A used as an immunosuppressant agent in organ transplant patient. Okay, so if someone needed a kidney or some other part of essential vital organ, and if a vital organ is transferred from an individual to patient, that time patient's body sometimes show immunological reactions, because our immune system can analyze what is self or what is non-self. So that time, although doctors before the transplantation, doctors has gone through so many type of tests and other things, but after that also that person that which mainly involved in this type of organ transplantation procedure, that has to taken immunosuppressant according to the guidelines of doctors. Okay, so this immunosuppressant agent cyclosporin A is also isolated by the fungus, that is Trichoderma polyosporum. Another bioactive molecule is statin. This statin is also produced by yeast, Monoscus purpuris. And this statin is used as a blood cholesterol lowering agent. It acts as a competitive inhibiting the enzyme responsible for the synthesis of cholesterol. So, statin is used as a blood cholesterol lowering agent. This is again isolated. This is again produced by a yeast strain. Okay. So these are some household or industrial products, which is isolated from various fungus, yeast, or bacterial strain. I hope all the content has been cleared now. If you have any doubt related to this particular lecture, please let me know. We will discuss it later on. Till then, take care, bye bye, and keep learning. Namaskar.